Bom dia a todos. É um prazer grande estar aqui de novo com vocês. Mais um mês que a gente tem um seminário aqui do Ciência em Cena. E hoje a gente tem a presença de uma convidada internacional e a gente gostaria de aproveitar essa oportunidade para agradecer o convite, né, assim, agradecer a presença dela aqui. E também eu gostaria de agradecer a presença e a participação dos nossos colegas, a Nicole Moraes e o Guilherme Furlan, que estão dando apoio aqui nos bastidores da plataforma para a gente ter esse evento bem sucedido novamente. Tá? Então, hoje a gente tem a satisfação de receber a Laura Natália Gonçalves Garcia. Ela é coordenadora do Hexacor, Facility de Bioinformática e Análise de Dados para Computação Científica e Serviços Tecnológicos para Pesquisa da Universidade de Los Angeles. Ela é mestre, ela é bióloga pela Universidade de Los Angeles, é mestre em bioinformática, uma das primeiras mestres formadas em bioinformática na Colômbia. Até a presença dela aqui é graças ao contato do professor Diego Riani. E a gente tem o prazer de tê-la aqui. Atualmente, ela também está fazendo um doutorado na Universidade de Montpellier, na França, e também um joint doutorado na Universidade de Los Angeles, na área também de análise de dados. Então, hoje ela vai falar um pouco da experiência dela de pesquisa. Já faz vários anos que ela trabalha com bioinformática e análise de dados, e hoje ela vai falar um pouquinho dessa experiência dela, experiência profissional, experiência científica e dos resultados científicos que ela vem adquirindo. Então, Laura, muchas gracias por aceptar participar con nosotros aquí. Y es una satisfacción muy grande. Yo te conozco ya hace un rato y para nosotros es muy bueno tener una invitada de afuera también, incluso para mostrar a los estudiantes de posgrado de nosotros lo que es posible hacer y el impacto que tu profesión de bioinformática vem haciendo en tu país. Muchísimas gracias. Nicole, muito obrigado. Guilherme, muito obrigada. Agora eu vou pedir para a Laura apresentar, colocar a apresentação dela, e a gente vai sair de cena e deixar, então, a Laura falar um pouquinho para a gente. Laura, muchas gracias nuevamente. Por favor,
Muchas gracias, Flavia. Well, so I'm going to speak in English now. So thank you, Flavia, for your introduction, and thank you, everyone, for joining me today in this presentation. So, as Flavia was saying, I'm a PhD student at Universidad de Los Andes, Colombia, and in Université de Montpellier in France. And the idea here in my PhD is to join bioinformatics and computational biology, developing different kind of algorithms and software, and apply them into different organisms. Currently, I'm working with Leishmania parasites and also with coffee crops. So today we are going to talk about how we can use k-mers to solve different genomic problems and how we can use them also to reconstruct the pangenomes. Well, first of all, it's important for us to understand what is a pangenome. So, according to the first definition of Tetlin in 2005, the pangenome is a catalog of all the genes present in each species or isolates and the order of the genes within the genome. Currently, we have different approaches that also include intergenic regions, transposable elements. We can also include different kind of genomic features among the genomes. But for this presentation, we are just focusing on the first definition. And to give you a small example, if we have here three genomes that are called A, B, and C, and each of these color blocks is representing one gene within each genome. The idea when we reconstruct the pangenome is to identify the genes that are orthologous genes or that belong to the same gene family among the genomes, and then use them to build this kind of a representation with the order of the genes that are present in all the genomes that we are including. We can also represent this pangenome as a matrix of presence absence of the gene families, and based on that, we currently classify them as core genomes, or the genes that are present in the majority of the genomes that we are analyzing, and the accessory genome, that is mainly those genes that are unique for one genome or are present in some of them but are not uh, the base of the species that we are analyzing. So basically, to reconstruct a pangenome, we need first to have high quality data, and there are three important steps that we have to solve before we start constructing the pangenome. The first one is that we need high quality genomes, that we need assemblies that are super curated and contiguous, just to understand the order of the genes that are in them. Then we also need that those genomes are annotated, both with the transposable elements that are going to be TEs in this presentation, and also the gene annotation. Both of them are important to reconstruct, to annotate in the genomes. And after that, we can analyze the gene families that are present in the genomes, and finally we can get this pangenome. So in this presentation, we are going to approach in each section one of these challenges that we have when we are trying to analyze pangenomes. Well, so first of all, we are going to talk about genome assembly. The challenge of the idea here is to assemble high quality genomes that are contiguous, and we are focusing in the assemblies using long reads that can be reads generated by Pacific Biosciences, especially the hi-fi long reads, and also we can use the nanopore long reads. So to give you some of the context, when we are analyzing these long reads, one of the traditional algorithms that we use is called the overlap layout consensus. And in this case, we have all these reads that are color coded for this example, and then we need to identify all the overlaps between each pair of genes. Like here that we have the color coded, the orange is overlapping with the other orange, and then with this R9 read here. And after that, we can reconstruct a layout that is going to have all the overlaps and the possible paths that can join the reads and the order of them. For that reason, we have like these arrows here in the layout. And finally, we are going to reconstruct a path that is going to be our genome. And this path in this case is called a Hamiltonian path. And the idea here is that we reconstruct just one path that goes for each node one time, and it's gonna pass for all the nodes or all the reads, and finally with that we are going to have the consensus sequence. What is the problem here that we are addressing is that the identification of the overlaps requires an all versus all comparison of all the reads that we are going to analyze, and that problem is sometimes is inefficient in terms of the computing resources that we need because of the search space that's going to be large. So one of the approaches that have been developed is the reduction of that search space using minimizers. Well, what is a minimizer? So the idea of a minimizer is that if we have this S as a read or a sequence, we can split that S in overlapping windows or subsequences of a fixed size. In this case, all those windows or lines that we can see here. And for each of those windows, we are going to select the best k-mer or the best substring of a fixed size again. And then we are going to represent the entire subsequence, so the entire substring, using that small k-mer that is going to be the minimizer. And the idea here is that contiguous or neighbor windows are going to have the same minimizer representing them. And at the end of the process, all the S or all the sequence is going to be represented by a small set of minimizers. And then when we are comparing the overlaps between each pair of the reads, we are not going to compare the entire S or the entire read, but just the small set of minimizers, and it's going to be easier in terms of the computing time. Well, so the idea here of the algorithm that we have developed is that we design a new function to identify or to pick the minimizers for each of the sequences. So the idea here is that we have all the k-mers of a fixed size. In this case is 15, and we have the distribution of the k-mer frequency in the reads that we are analyzing. And we have a ranking function that is based on the mode of the frequency. So we are going to give lower values or lower ranks for the k-mers that are close to the mode, and larger values for the reads that are far from the mode. And this is going to help us um, to give more importance or more value to these k-mers here in the distribution that are giving us a lot of information, and less value to those here at the beginning of the distribution that can be related to sequencing errors or rare k-mers within our genomes. And also we are going to give large values to these k-mers here that are going to be mainly repetitions and are not going to help us a lot in the reconstruction of the genome. So then we are going to compare the minimizers between each pair of reads, and based on that, we are going to reconstruct our genome. So after that, the idea here is that we have different metrics as the number of overlaps that we have, and the size of the overlaps. And with that, we develop also a machine learning approach that is going to be trained with the safe edges that are defined as the edges that are reciprocal hits between pairs of reads. And with them, we have the distribution or the metrics that we are going to use for the algorithm. And using those metrics, then we can compare all of the edges that are not safe, that are candidate edges, and pick the edges that we are going to use to reconstruct the path. So finally, we are going to have this path with the bold lines that is going to be the genome that we are reconstructing. So with this approach, we perform different benchmark experiments. Here you can see some experiments using PacBio HiFi datasets for the rice, using reads of 15 kilobase pairs, 20 kilobase pairs. We also have some maize reads and reads from the human cell line CHM13, that is a haploid cell line. And with the section A of this figure, we can see that our assemblies that are in blue, the circles in blue, we get better contiguity than the assemblies that we can get with fly for all the cases. And in some of the cases, especially rice, we can get a similar contiguity that the contiguity that we have with HiFi ASM or Canoe. um, but also we are faster than Canoe. That's one of the big advantages of using our software. In the section B of the figure, we can see the number of misassemblies or errors that we have when we are assembling the genomes, and in this case, we are also in the blue color, and we have few misassemblies that the number of misassemblies we got with hi-fi ASM and Cano in the majority of the cases, especially implants. And then if we see the results using nanopore reads, we also see here that we have better contiguity than Fly, in some cases than Canoe, and also NECAT, that were the software that we were using for this benchmark. And also, depending on the case, we have fewer errors with these assemblies. One important thing here is that we are focusing in low error rate reads. So for these nanopore experiments, we were using the first nanopore technology reads that is between 10 and 20% of error. So we have to perform a correction step of the reads. In this case, we were using NICAT as a tool to correct the reads. We tried different tools, but NICAT was the tool that worked the best for us. So for this case, if you are going to use old nanopore reads, it's useful to correct them first with the cut before using NGSEP to perform the assemblies. Well, and then finally, we can also perform a polishing step if you have short reads. In this case, Illumina reads that are the reads we are using for different projects. And the idea here is that with the long read, especially the nanopore reads, we are going to have different errors within the assembly, and we can use the short reads that have less errors to correct or polish this genome. In this case, the idea is to use one software or one tool to map all the reads. We can use Bowtie, BWA, or in this case, we are using our own tool that is NGSEP Reads Aligner. And with that, we can align all or map all the reads to the genome, and then we can use a software to identify variants. In this case, also, we are using our own tool, NGSEP Variance Detector, to generate that matrix or of SNPs or indels or variations in general that we have. And finally, we can reconstruct that genome to remove the majority of the errors that we can have. And then finally, it's always possible to do a manual curation step to finally map all the long reads, the short reads, compare with reference genomes, and manually curate the genome to have it in the better quality that is going to be possible. So at the end of each section, I'm going to give you here two QR codes. One is our SourceForge QR code if you want to download the last stable release of the software. And also here is our GitHub QR code where you can download the most recent version of the software with all the changes that we are doing like every day. Well, assuming we have high quality genomes and we are satisfied with the results we got, then we need to perform the gene annotation step because we need to get all the genes that are present in that in those genomes. However, if we have larger genomes or regions that are very complex or with a lot of repetitions, it's important to first annotate the transposable elements in there to also perform in a more efficient way the gene annotation step. Um, well, so why is this important? This is important because in plants, that is the organism that I work the most, we have between 10 and 90% of transposable elements as the total content of the plant. And if we are going to annotate the genes, it's useful if we just mask these 90% of the genome before we perform the annotation step, to avoid the tool to try to identify a lot of genes in regions that we are not going to have them. And also in this case, we have the same problem as in the genome assembly because we relied on all versus all comparisons that are based on blast applications, and the most used tool for that is repeat masker. So when we saw here that the problem was similar to the genome assembly problem, we asked ourselves if we can use the minimizers instead of the last all versus all comparisons to perform the same task. And well, the answer here was yes, we can do that. And we developed an algorithm to identify or annotate these transposable elements in our genome. So the idea here is that we also are going to have the genomes and some um, transposable elements databases. That's one important thing here is that we need a library to do a search for an homology search uh, in this case. And then we are going to represent the genome as a set of these minimizers, or we are going to index the minimizers in those genomes. And we are going to do the same with the transposable elements database, as you can see here. And then the idea, as the same case of the genome assembly, is that we have the complete genome represented as a set of minimizers. Each transposon in the database is going to be also represented as a set of minimizers. And comparing just the minimizers, we can align them and annotate that. And then in the output, we're going to have for the genome the position, the chromosome first and last position, the family, and the specific element in the database that has a match with the region that we are annotating. This family depends on the annotation of the library. So if we here know that it's a LTR copia, then here we can say it's an LTR copia. But if here you just say element, then the family is gonna be element. We're not going to have that much information. So that's the idea behind the algorithm to perform this annotation. So we performed two benchmarks for this case, one using the Altior retro elements because those are the elements that are present in the majority of the plant genomes. And if you can see here, we achieve good precision and sensitivity in the majority of the cases. So here we were using the output of Impactor DOS, that is the software to annotate the novel LTR retro elements within the genomes. And so using that libraries, we got good annotations of our elements using Arabidopsis, some wild coffee, in this case, Coffea humboldtiana, and the rice that is Oryza sativa. And well, if you see here for Arabidopsis is over 0.75 for precision and sensitivity using that database is over 60, 0.6 for coffee, and over 0.75 again for the rice. And the huge impact here is that we are reducing the computing times a lot. So if we compare ourselves with the repeat masker, that is the traditional approach, we can annotate these elements in seconds in comparison to the time that repeat masker requires. So this precision and sensitivity, that's one important thing, is calculated in comparison to the repeat masker outputs because we don't have gold standards to do this comparison. So those are the numbers that we got in comparison to the outputs that repeat masker was reporting. And that's the result for LTR retro elements. In the case of a full transposable element annotation, we were using different libraries. So we have an Arabidopsis specific library, a coffee specific library, the REP-based library, that is the database that is used in repeat masker. And we also have this TrepDB that is mainly used for like for wheat or rice or different plants, and but now it has a lot of different species. And as you can see here in the Arabidopsis, the database that works better is the specific Arabidopsis database. In the case of coffee, is the coffee specific database, and in the rice are those rep based on RepDB that are the databases that contain more related elements. So the idea or the main result here is that if we are using closed or relatively closed libraries, we can get good annotations in the organisms that we are comparing, and these results are using two iterations of the software. So we perform a first annotation, then we use that annotation as the input to perform a second round of the annotation, and that's the output. So in all the cases, if we increase the number of iterations in the annotation process, we are going to have better sensitivity values, and the precision can be reduced a little, but we are going to have good sensitivity values for that. And also in this case, the reduction of time is super important and huge for this process. So finally, if we are using one of the complex genomes that we have, that is the hexaploid with genome, we can see here that the 85% of the genome is composed by transposable elements. And if we try to use repeat masker to perform this annotation, we are gonna need months and huge computing capacities to do that annotation. With our software, we can do that annotation in less than 2 hours per chromosome that we are analyzing, and using less than 250 gigabytes of RAM memory per chromosome. So this has a huge impact in the process of gene annotations, because we don't need to wait for a month to get the transposable element annotation before annotating the genes. We can just work for one day in the worst scenario to get all the annotation, and then we can continue with other steps. And if we see here the distribution of the transposable elements that we got for this with genome correspond to the annotations that are in the genome now. So it's mainly LTR elements and DNA cacta elements, as was expected. So the annotation is working well, and we are going to be happy if you try it and tell us how it works for your organisms. Well, so here again the QR codes, and finally I'm going to talk about the ortholog grouping, or the last step in the process. Assuming we have again the high quality assemblies, and we have all the assemblies with a good quality annotation as well. And in this case, we also have the same problem. The identification of ortho groups or gene families is based on all versus all comparisons, and is also a demanding step, and it requires a lot of time and computing resources. And so, as a context, here again, looking at the colors as the first slides, we have our three genomes, A, B, and C, and those are color coded. And in this case, we also need to have the pairwise alignment between each pair of the genes, and based on that, we are going to organize them into gene families or orthologous groups. Well, so our solution here is to identify all the gamers that are present in all of the genes that we are analyzing. And we also have according to the distribution of the frequency of the gamers, we have them ranked. And we also have, like, in which of the we have an index to know each gamer in which gene was identified as is seen here. So at the end, with that information and these schemas that are shared, then we are going to identify some edges between each pair of genes, and we are going to have a graph of the relations among the genes. So here we have like the this gene family, this other, and this gene. Then we have the complete graph. And with that, do we perform two different algorithms. The first one is connected components that is going to reduce the graph or identify those sets of genes that are highly related. And but we can have like here some edges that are not real, uh, but are there because they share a lot of gamers. So after this connected component step, we perform some Markov clustering steps that also can be modified to finally get the gene families or the orthologous groups. So in this case, green corresponds to one orthologous group or gene family, the yellow one is another, and the blue one is another. And these are going to be the resulting families that we can use for to reconstruct the pangenome or align the genomes. So I'm going to show you how we can perform that. Well, so the first approximation of the first output as a pangenome is going to be the presence of absence matrix. So in this case, we have for each of the gene families a matrix representing the 1 or 0 according to its presence within the species. So in this case, the first family is present in the three species or genomes we are comparing. So we have 111. In the case of the yellow one, is just in two of the genome, so it's 110, and the same for the case of the blue one. And with that information, we can calculate gene frequencies and identify what's the core genome and what's the soft genome for each case, the core and the accessory. Sorry, and also we have a threshold if you want to fix it to like 90% of the 90% of frequency is okay to consider that as a core genome. So you can also modify that parameter to identify what's core and what's accessory within the genomes you're analyzing. And finally, we had here an implementation of the tag chainer algorithm to align the genomes based on those the presence absence on those genes, and also identifying the blocks of synteny across them. So to see some of the results of our clustering, as you can see here, we are comparing um, some genes of the bilateria and also some of the genes from mammalia. We have here the results comparing our software against OrthoFinder 1 and OrthoFinder 2. That is also a commonly used software. And in the case of bilateria and mammalia, we achieve good values of recall and precision as well, being better the results in cases such as mammalia that we have a closer taxonomies in the analysis. So it's really useful if we are including in the analysis organisms, species, or just genomes that belong to that are closely related among them, and that's because OrthoFinder relies in the phylogeny of the species that are going to be compared them. So it works well when we have species in all the like ranging for all the taxonomy, but it's not as good when we have closer species. If we see in the terms of pangenomes, as you can see here, our algorithm is stable in terms of the core genome. So here we have the soft core and the exact core. The soft core is using a threshold of 90% of the frequency of the gene family, across the isolates, or well, in this case, is are isolates because we are comparing bacteria. So the core genome is stable when we are adding more and more and more genomes. So that's a good result. And the accessory genome is going to be increased when we increase the genome as is expected, because we are going to have more unique genes in the process that are present in the different genomes that we are adding there. And also, if we compare ourselves with one tool that is available, that is Pangolin, we have similar values for the core genomes, and we have some differences in the accessory genomes because the process of identifying the gene families works better with our algorithm, so we are not overestimating the number of different families when we include more genomes. Um, and finally, here I'm showing you the genome alignment of Orisa sativa, that is here with the chromosome numbers, and Brachypodium distachyon here. And as you can see, with this information of the gene families and the synteny algorithm, we can identify the blocks of synteny that are conserved in both of the genomes that we are comparing, and we can reorganize the genome as an output of this software. Well, so here again, the QR codes for the SourceForge and GitHub pages for us if you want to give it a try. Um, and well, finally, in conclusion, the main point of this presentation is that we can use k-mers in several steps of pangenomics and genomics reconstruction, and there's still a lot of space for developing and improving the tools, so we can continue developing different approaches, perform more benchmarks, being more specific to, in the case of transposable elements, like just developing different tools only for LTRs, or say for each family in a specific way. We can continue working with the facing deployed assemblies, different kind of information, and also in the field of the pangenomic reconstruction. If we want not only to have the present substance matrix, we can continue developing tools to get the pangenome in a graph representation, to map reads or perform different tasks in the in genomics. Well, so finally here I have the complete articles where you can find more information about the tools that I was talking today. So here we have in molecular ecology resources the NGSEP for release of the software. In this release, there's the information about the synteny, the genome's alignment, the orthogroup identification. And we have also this second paper here that describes the algorithms for genome assembly using long DNA sequences, in case you want to read them all. And I hope this semester the transposable element manuscript is going to be also published, so you can read it. Well, the computer resources that we used for this research are the resources from Universidad de los Andes, that has the core facility that I'm leading, that is Exacore. So we have in this core facility all the computing resources like HPC cluster, virtual machines, cloud computing resources that are available for the community if you want to use them. So you can visit our webpage here, exaccore.uniondes.edu.co slash EN in case of English, or it's also available in Spanish. Or you can write us an email to Exaccore, so we can work together and see if we can help you with your research projects. Well, so thank you everyone for joining this presentation today. If you have any questions, I'm here to answer them. And also, if you want, you can write me an email to my Uniandes or my IRD emails. And here again, our QR codes if you want to download or give a try to our software. Thank you so much.
Um, Thank you, Laura. Yeah, I think your presentation was really nice. I really love this topic, and your slides are really great. I want to do that. But your presentation is really nice. We have some questions for you now. I will start with the uh, multiple question here from Professor Diego. So I think Diego is very happy to have you here, but he's asking some more difficult questions here. I think you need to answer him. So Diego is telling us, congratulations, your very nice talk. And he's interested to know how do you pick camera minimizer for genomes of diploid or higher ploidy organisms when you have multiple peaks in camera spectrum, and which peak to use to rank the minimizers?
Well, in the case of diploid genomes, we perform the same approach as the original, like the haploid genome. So we use the same set of minimizers and the K-mer spectra. However, at the end of the first assembly stage, we use our tools that were developed for building variation databases, and with that, we perform the phasing of the information. So it's like another step included in the assembly process, but we use the same distribution for the gamers and minimizers in this case.
Okay, I hope Diego is fine with that. I have a question because I'm working with other communities of organisms, and I was thinking you showed that your tool is very useful for assembling the genomes, but I'm interested, for instance, if we have a very complex community of different organisms with smaller genomes and a little bit higher genomes, like a phytoplankton community, what happens if we use your tool? Can we assemble it as easy as you show it for bacteria, or then it starts to be more complicated story?
Well, in this case, I think you're talking about metagenomes, right? Well, I
Yes.
don't know. The answer is I haven't tried that. So I have to try and I can tell you if that works for metagenomes. So with bacteria, just one bacteria, it works fine, but I'm not sure what happens when we include more than one bacteria in the same sample.
Because I saw that in your analysis you have the core genome, and you have also soft genome, so different genomes that you can have afterwards using ortholog analysis, right? So
Yeah.
do you think this kind of comparison or this type of alignment could be useful for something like that to distinguish between different organisms in the same complex community, or it's not the way to go?
Well, um, if you have a set of all the genes that are in the community, you can run the algorithm to identify the gene families and orthogroups. The problem there is that we need previous information to know each gene, that if each gene belongs to one or another species. If not, you can have the clusters. The clusters are going to work fine. You can have super fast all the clusters, but then you have to assign each of the genes for each cluster to one of another species to know them.
Thank you. We have another other questions here. Again, Professor Diego. For the detection of ortho groups, user will have two important parameters to set: how to weight cameras, and the inflation value of the MCL clustering. How to choose or to set these values?
Okay, there are different parameters in the software here. I think the MCL clustering, the inflation value is by default. Um, how can you choose them? I think we need to perform different experiments to know the best value, those best values for the analysis we were using. So in different cases, we saw that sometimes with just the connected component algorithm, it works fine identifying the groups, but it depends on the genes that we have to know if we need to perform the Markov clustering or not, and the value of that inflation value and the configuration of the Markov clustering step, and how to weight k-mers. We here can know the size, so we can fix the size of the k-mer, and the function is a default function that we have implemented. So you can just fix the length of the k-mer that you are using there.
Okay, I will just jump to another question here, then I go to a final question from Diego. So, Percy Vergara, thank you, Percy, for your participation. He's asking here, en el caso del elemento de transposición, funcionaría para organismos antiguos como los tripanosomatídeos, ya que una de las deficiencias son las librerías disponibles. ¿Cómo enfocar esos problemas?
Well, I'm currently with the same problem. I'm annotating the transposable elements in Leishmania. So yeah, I know we have a problem with the available libraries. So I think the here, well, we don't have a lot of LTR elements in the trypanosomatid family, so we can use with larger databases such as Rebase and see what happens and perform more rounds of iterations. And there are some articles that have the transposable element information of some leishmania I saw. I'm not sure about trypanosoma or another species. So you can join all that information in one library, or like the red base and that specific information, and perform different series of iterations of the software to identify your transposable elements. But yeah, the available libraries is a problem we have to deal with and iterate over that.
We have also some congratulations here from Professor Zagato. He liked your talk. Congratulations. He could not follow to the end, but he's saying that it was great. Percy also said it was excellent. So Diego has another question. Do you have some ideas on how to assemble high-ploidy and highly polymorphic genomes aimed at chromosome scale and recovering all chromosome copies? Would the minimizers help there also?
Well, well, it depends on the kind of data you have. So, my first idea is that if you have good quality data, such as PacBio hi-fi reads, it should be easier than if you have a high error rate reads or short reads. So, with that information, is going to be easier to assemble those genomes. And well, our minimizers approach, as I was saying, can perform that layout or the first layout reconstruction. So if the minimizers and the differences between the copies of those, like the each phase of the genome is clearly differentiated. So if we have minimizers that are dividing them, it's going to help a lot. So and then also we need different approaches for performing the phasing of those parts that are not that are not easy to separate using just minimizers. So just the minimizers help a lot, because if we are ranking them, we can use specifically those minimizers to separate one haplotype to the other. But we need more tools and develop new strategies in those regions that are not easily faced, because they are super similar.
Okay, thank you so much, Lara. I think we don't have the other comments or questions for now. But yeah, the presentation, we have some congratulations here for you. Anyway, the presentation will be available at the YouTube channel Ciência em Cena. And the chat will be also repeated there, so the people can also consult the questions and follow what happens in the questions and answers session. So for us, it was really a great pleasure to have you here. I think this year you are the first foreigner presenter in our seminar, and this is a great achievement for us. Thanks to Professor Diego and you for accepting the invitation. So I would like to thank all the colleagues that helped this happen again. So Guilherme and Nicole that are in the background here to help us in the development of this seminar and continuity of that. I thank you all people that are following us in the Ciência em Cena channel, and I hope you guys can enjoy the seminars, and please share with your colleagues, share the information that we have here to help our colleagues to do great research and have information about great achievements that the researchers are having around the globe. So thank you very much. And I invite the people to follow us in the channel and see the further seminars that we have. Don't lose our next seminar next month, hopefully. And then we are going to be happy to find you guys here again. So Lara, thank you very, very much. And this is all for today. Thank you. Bye-bye.
Thank you. Bye.